Okay, everyone, I think we're going to get going. So first of all, I think everyone will agree we've had an amazing conference so far, and we're here for an action-packed afternoon. And I am Laura Kiesling. I have the opportunity of chairing this first session of the afternoon, which is going to be short talks. And so I ask that if you do have a question, we'll probably only be able to take one or two. So please only ask one question if you have it, and I'm going to try to keep us all on time. Okay, so just to kick this off, I'm happy to introduce to you Laurie Boyer. Laurie is one of my great colleagues at the Koch Institute. She is a professor of biology and biological engineering at MIT. She earned her doctorate in biochemistry from UMass Medical School and then joined the Whitehead as a postdoctoral fellow in the Yanish and Young labs. There she pioneered the development of high-throughput platforms for genome-wide analysis of transcription factors and chromatin regulators in stem cells. This really opened the field of stem cell genomics. She launched her own lab in 2007, studying how gene regulation regulates cell fate decisions, including in heart development. Her work bridges developmental biology and tissue engineering with a focus on how DNA is packaged and remodeled in chromatin. She uses stem cells, mouse models, and engineered heart tissues, and her team is uncovering the genetic circuits that drive heart formation. So she is going to be telling us today about traffic control, high-resolution mapping of ADCs within the lysosomal compartment. Laurie.
All right, great. Thank you for that introduction. That introduction now is probably very confusing. How does someone go from analyzing gene expression networks and cell fate to mapping ADCs in the lysosomal compartment? Well, hopefully by the end of the talk, you'll see there are some parallels here. It's tools, high throughput, high resolution. This was a side project that became a passion project. So I'd like to start by thanking the organizers for giving me the opportunity to talk to you today about our work to develop a tool that allows scalable imaging of subcellular compartments, right? And this is very much still a work in progress. I'm not going to have any conclusions for you, but I hope you will come away with two messages. One, our platform is really great for imaging antibody drug conjugates or other nanomedicines within a complex cellular environment. And two, these maps that we're generating have very good value for understanding the behavior of these nanomedicines within cells and could potentially be used to improve drug efficacy. So as you've heard throughout the symposium, there are a lot of flavors of these antibody drug conjugates, right? A large proportion of them are designed to target specific receptors, such as the transferrin receptor, which I'll provide some examples for you today, and that by targeting these receptors, the antibody or the drug can piggyback on these receptors to get into the cell through primarily a process called clathrin-mediated endocytosis. Well, so that's one piece of the puzzle, one piece of the business end. The other is, how do you get this drug all the way to the lysosome for processing, if that's your target organelle? It's not so simple because the antibody drug conjugate needs to, once in the cell, navigate this complex network of organelles, or the endocytic compartment, right? And to make it ultimately to the lysosome, which is the degradative compartment that is important for payload release and therapeutic efficacy. So that said, in fact, only a small proportion of the drug ends up in the lysosome. It can be trafficked back out of the cell, it can get stuck in this compartment, or it can be degraded elsewhere. Most times we don't have that information. And that information is invisible to most scientists because of the small, complex nature of these compartments. So unsurprisingly, given that we need to go from here to here, and we don't know the in-between, understanding the process by which these antibodies can reach the lysosome by examining these compartments can help potentially tune development of antibodies to improve their targeting efficiency. So as I mentioned, it's a very complex network. It's hard to visualize. And as an example, I'm just going to show you here that commonly, we've heard from a lot of our colleagues that they've tried to resolve these nanomedicines within the endocytic compartment using fluorescence microscopy, but they run into problems. And the problem is the diffraction limit of light. So anything less than 200 nanometers will show up as a single point. So here, even though we get nice staining of early endosomes using known markers or LAMP1 for late endosomes or lysosomes, as well as an antibody or looking at the antibody itself, once you merge these signals, even though they are not actually overlapping, they appear such. So that's a problem, right? We need to be able to resolve where these antibodies are at any given time in the right cellular compartment. Advances in super resolution microscopy can overcome the diffraction limit. However, these instruments are very expensive, require expertise to use, and are generally low throughput. And so for an academic lab, that's pretty much off the table. So what we tried to do then is to find another approach, a simple approach that would allow us to deconvolve these signals without having to use specialized microscopes. So we turned to an approach that was invented by Ed Boyden about 10 years ago, which instead of using microscopes to look at smaller and smaller objects, he just thought, well, why not just blow up the cell? So by taking a cell or a tissue and embedding it in a swellable hydrogel, you can actually isotropically expand that cell by 4.5-fold, which is at the level of super resolution, and increase the volume that you can also visualize, and I'll come back to this later, effectively now mimicking super resolution, but by simply using a conventional microscope. So this was really exciting to us. We were using the slide-based approach, which was how it was designed in the lab for other projects, and we were just like, wow, these images are amazing. However, coming from the genomics background where we like a lot of data really fast in an automated fashion, I thought, well, how do we make this to the level of a screening platform? How do we go beyond a slide and manual imaging? And so we thought, okay, well, let's adapt expansion to a 96-well plate. That should be easy. Well, it wasn't so easy, and it took us years to do it, but we managed. So what we ended up doing is developing a device, as shown here. I don't know why it's twirling so much, but essentially this design has an intelligent design where it can pick up a small volume of gel solution, deposit it on cells within a 96-well plate, and the gel polymerization is catalyzed by UV light, and then water is added to swell the gel. Okay, so now we have a plate. Oh, and I just want to show you the tips of the posts have been designed very carefully to have these reticulated grooves so that we can pick up precisely and deliver precisely a sub-nanoliter droplet of the gel solution to the well to allow it to fully expand without hitting the sides of the well. Okay, that said, now we have a platform that is compatible with high content imaging. As shown here, we typically use the Opera Phoenix. I'm just giving you an example of the type of data that we see. Here's a plate of cells that are stained for alpha tubulin. Here's one well and a field A view shown here. And the reason why I'm showing this, you can see beautifully that we have nice, good staining, and we've done a lot of validation to show that we achieve the same or very similar resolution to the initial approach. So I'm going to be showing you some examples today of fields of view, just for your reference. Okay, so how did we do? Does our platform work now to help resolve the ADC within these various organelles? So we used an anti-human transferrin binder, or an antibody, that was provided by Sanofi. We treated cells, and after 30 minutes, we stained and expanded them. We always take actually images pre-expansion and post-expansion. It helps us to validate the information. So in this case, you can see, as I showed you in the previous slide, that pre-expansion, you just kind of get this big blur, right? And you can't really tell where the antibody is located. But on the right, you'll now see that we get much better resolution of each of these compartments, and that when you overlay these, we can now start to more confidently assign where that drug is relative to an endosome or a lysosome. Okay, so we're pretty happy with those results, and so now we wanted to ask whether or not we could resolve the behavior of certain antibody drug conjugates over time, and in comparison to other antibody drug conjugates. Just for really demonstration purposes, I'm showing you data from an antibody. It's the same antibody. One is not conjugated where the other one is. So these are both anti-TFR antibodies. Again, these are provided by Sanofi. And antibody 2 here is conjugated actually with acid alpha glucosidase, or GAA as I call it. And the reason why we were pretty keen on using this antibody is for a number of reasons. One, GAA is a critical lysosomal enzyme, and so it should show up in the lysosome. And this antibody was also modified to recognize a receptor on the lysosome to ensure more efficient targeting. So as you can see in this zoomed in image here, the yellow being the anti-TFR and the magenta being LAMP1, in the conjugated antibody, or the GAA ADC, we see this beautiful co-localization of the ADC with lysosomes. And this is after 5 hours, which we would expect a significant proportion of that ADC could reach the lysosomal compartment at that time. In contrast, we pick up both signals, but if you look closely, they don't actually overlap that well, right? So it's consistent with what we thought we would observe, that this antibody should be better at co-localizing over time with the lysosome than the unconjugated antibody. And because we can do this at scale, we've looked at a number of different time points and conditions, and now we have enough data to use more sophisticated algorithms, such as co-localization by cross-correlation, which was developed to look at super-resolution data. So essentially, this method computes the correlation between intensity signals of these two channels, in this case, and by spatial offsets, right? So essentially, a 0 value is direct co-localization, where variance from this 0 value is actually suggests spatial relationship at varying distances. So I hope you can appreciate from these data that across our 5-hour time point, antibody 2 does pretty well, right? So you can see within 2 hours that almost all of it, at least the signal in the total antibody signal that we can detect in the lysosome is in the lysosome. And you might see this bump here, but it was really just because the design of the experiment, we didn't kind of pulse chase, so it may be just residual antibody that gets into the cell later. In contrast, our antibody 2 does not show that same correlation. So I want to just point out, even though I'm not showing you these data, these differences are not discernible if you just use conventional fluorescence microscopy. You would call both antibodies completely overlapping the lysosome, right? And so you need this higher resolution to make these distinctions. Okay, so we need ground truth, right? In the interest of time, I'm going to go a little bit faster. So one of the things we wanted to do, we're using antibodies. You see what you see, but you don't see what you don't see. So although we're getting great resolution, we don't get context, right? And what are our antibodies actually recognizing? We know that there's a lot of spatial and morphological heterogeneity in these compartments. So this is just a demonstration of what we've been doing lately to kind of provide more ground truth. So we've been using with the KI nanocore a process called CLEM. So basically this is correlative light electron microscopy, and then it enables detailed morphological resolution of the lysosomal compartment. I just want to point out a couple of things here to you that you may or may not see. Essentially, we can stain our slides with the antibodies I just showed you, and then the samples are processed for EM. You can then overlay those images to get at the fine resolution of where that drug or where the is within the cell. So here in red is LAMP1, the ADC is green, and as you can see, at early time points at 30 minutes, at least in this example, we don't see much co-localization, which is what we would expect. On the other hand, at 5 hours, we see a lot of these really nice structures where we see direct overlap between the ADC and LAMP1. So I'm going to skip over what we saw for the other antibody. The only thing that I'm going to mention is that in many cases, the antibody, we don't know what it overlaps with. 100% of the antibody doesn't go to 100% of the organelles. So where is it going? Right, this approach is at least now allowing us to identify some structures in the cell, to give us some context, to develop hypotheses for where it might be going if it's not going to these compartments. And that information, I think, is going to be valuable later on. All right, so last slide. So now that we can generate a lot of data at high resolution, we can also get volumetric measurements because we have increased axial resolution as well. And why is this important? So volume is actually interdependent with morphology, right? So I showed you at the beginning that the endocytic compartment is very morphologically heterogeneous. And we're kind of limited by antibodies. So sort of our thought was, like, if we could better understand and map morphology, the morphology space, and also overlap with what antibodies we do have, we can use sophisticated algorithms to start to output sort of some patterns, right? Here's just one demonstration of what we can do with organelle volume. We have a lot of data. There's 5,000 or so lysosomes in each of these time points. And I don't know if you can see this in this image, but essentially we can then overlay the signal of the antibody for each lysosome. And you can see at the 5-hour time point, we see a lot more antibody drug conjugate overlapping individual lysosomes. And we think, like, mapping these things for individual lysosomes is going to also provide a lot of key information, because we can take these morphologies and map them back onto our images to get a better understanding of how the ADC is distributed over time. So in closing, although a lot of this work is ongoing and we've started to make really good traction with machine learning models, sort of our pie in the sky goal here is to create these high resolution maps, right? Both using morphology, antibodies, agnostic stains. And by doing so, we hope to be able to kind of create this map where we can overlay the antibody signal for whatever your favorite nanomedicine or ADC is, and understand where that drug is being targeted at any time and at specific organelles during the process in the cells. And so we think that this will help us find sort of where the drug might be getting stuck. It might help us define where that drug is being more targeted. And at the end of the day, we think it will provide really informative and important information for being able to tune or select drugs that may have more efficacy. So with that, I will end there, and I'll take any questions. And I'm also happy to talk to people if you think our platform could help you for your own studies and questions, because we can resolve lots of different structures in the cell. And I want to thank members of my lab, especially John Day, who developed the platform, the KI Nanotechnology Core, Abigail and Giovanni, as well as our Sanofi collaborators who provided us with some of the materials and guidance. So with that, I will take questions. Thank you.